Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
It's my honor and a privilege to introduce our speaker today. We are receiving a talk from Dr. Hui Wang, who is a postdoc in the lab group of Hangzhou at UCLA. He's 2 years into his postdoc working on TomoNet, which is a tomography particle picking software that's popular for structures that have lattices as part of their biological feature. He has a background in computer science and then was accepted into the bioengineering department at UCLA before moving to structural biology. So in a moment, I'll ask him to start his talk, but I just want to say briefly that if you have any questions throughout the talk, if you want to drop them in the chat, please drop them in the Q&A section so that we can address those at the end of the talk, or otherwise wait for the end of the talk and then you can ask your questions in person. So with that being said, Dr. Wang, if you'd like to take it away.
Okay, thank you so much for the introduction, Alice. So let me share my screen. Okay, can you see the screen?
Yes, we can see your screen.
Okay, so let me see how I can. So yeah, hi everyone. This is Hui from UCLA in Dr. Hongzhou's lab. So it's my great honor to present you my my recent work about TomoNet, which is the title is automatic particle picking of flexible lattice-like structure for CryoET. So so this is kind of like the general content that will be present today. So I'll give some brief introduction of the overview design of the motivation of TomoNet, and then I will mainly focus on the two major particle picking tools in TomoNet. So for this particular webinar, I'll just assume that you all have a kind of like a general understanding of cryo-EM. So basically, this is another version of cryo-EM called cryo-electron tomography. Basically, I'm using this parasites flagellum study as an example. Basically, you just after you culture the cell, you extract the components you wanted and put them on the EM grid, do some screening to optimize the concentration and purification protocol. Then you load it on the electron microscope by tilting the sample from a certain degree to another certain degree. You get a tilt series. Using that tilt series, you can do a 3D tomogram reconstruction. As shown here, you can see a 9+2 axon structural construction with the resolving power. You can see some of the features around 50 nanometer, like this dining arms and those radio spokes. Beyond that, you can further do some sort of more averaging to basically the subtomograms, also being called particles, which will be referred to in the following talk. So those subtomograms are like oriented in different way and located different position. They're being extracted out and then align them to a reference like that to first eliminate the missing wedge artifacts. Basically, you have the missing wedge at different location, then average them together to eliminate the artifacts, and also by averaging, you improve the signal to noise ratio. Then ideally you could boost the resolution of the raw tomogram from 50 angstrom to a average map like better than 20 angstrom. So basically, this is what you would expect by averaging few hundred those 96 nanometer averages. You can see those kind of like the red is the inner dining, the cyan one is the outer arm dining. So there's one resolution limiting factor, which is the particle numbers, right? Based on this is not might not be really accurate, but this is based on my practical experience. Like for some structure, your target goal is between 12 to 25 angstrom resolution. Basically, you can see those globular domains. Then you may not need too many particles, but for the target resolution landing like a 5 to 8 angstrom, basically allow you to the secondary structure like the alpha helices and some of the shape of the beta sheaths. You probably need typically at least you need more than 1K particles, and specifically if you want to reach to like equivalent resolution to single particle, you need typically more than 10K particle to be able to see all the side chains to enable a accurate atomic modeling. So nowadays people are more kind of like want to push into the kind of like a higher resolution part. Another needs for so the more if you want more particle, which means you need to collect more a larger data collection, so which could result in hundreds of tomograms. So therefore, there's a need for large tomogram data set management, right? So this is just a screenshot of like how you just LS like all your tomograms that could be like a huge like a heavy use of terminal operations, and you barely don't have the visualization of each individual of your tomograms, and also for a entire pipeline of the CryoET and the subterranean average, you typically need to use a lot of softwares. Like then you certainly also need to do some conversion between software packages, like you kind of like convert from iMod convention to rely on for your 3D refinement. So basically, the TomoNet motivation to create TomoNet is basically address these two major challenges, right? So first is we kind of like to have a integrated workflow. Basically, we just put them in a seamless kind of like a pipeline. So we integrate the all the existing good to use software, and we put them in a single like interface, so you can just use them by click without you do the conversion, like switch the software like using some customized code. And the second is we kind of like implements two like software particle picking tools for more efficient or accurate particle picking. And this is so generally the TomoNet is a GUI based. So this is one of the major GUI for TomoNet design. So on the left part is the main module, like major functions. So you can start from the motion correction, 3D reconstruction using either iMod or RTOMO, and CTF estimation using CTF find, and you can do some ISONET mismatch correction in the ISONET tab, and also you can do some manual picking using the iMod function. And you don't need to really kind of start from the very beginning. You can you can kind of like chip in basically on whatever you already selected. For example, if you already reconstruct the tomogram somewhere else, you can directly use the ISONET function here to kind of like they're mostly independent. And another kind of like, I think the thing is good for the user is this table view of your tomograms. Basically, you can kind of have some direct operation or tracking of on your each of your tomograms. Like I have this column, so you can make some notes on them to indicate some like keywords or key element things you want to highlight for each of the tomogram. And you can also view them and do some action to each individual of them without using the terminal uh, uh, terminal commands. And okay, I guess I will be this is the part I want to highlight in this talk. So there is mainly two particle picking automation tools in TomoNet. So on the left is a geometry base, which is lattice-based function called expand. Basically, the idea is you use a few certified particle as the expansion center. So you kind of like following the lattice or the geometry of your sample to finally kind of like in this example to exhaust all the possible location for on your lattice. Another one is a neural network based picking. Basically, is more kind of based on the appearance of your particle, like is a feature detected strategy. So and they have their uh, uh, some more kind of like this is uh, on the geometry base, right? It's gives you more accuracy and better control because it's like you restrain your particle picking in a specific way. But the drawback is that it's kind of like likes your particle to form a lattice-like structure and require more user input. On the right part, it's the worst part is that the accuracy is typically lower and the way to exclude false positive is a little harder to control. But 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 but, but on a good side is that it's more generalized and require less input from the user and typically don't require any like a distribution on your particle. So I'll be explain this expand function first. So given that the lattice structure are actually common in biology, is kind of like a very economic strategy for the for them to kind of form some like a economic strategy to form a large or repetitive structures. For example, the virus capsid, right, and the microtubules, basically they're made of those tiled tubulin, and also the nuclear egress complexes, and also some surface layer of the both bacteria or archaea, and this is some also 3D paracrystallized PFR structure that attach to the flagellum. So since there's kind of like a strong needs for lattice structure particle picking, so then this is our kind of like a strategy of the expand, right? So you can imagine that this virus-like particle of HIV is a kind of like connected lattice. Basically, you can spread them out, kind of like have a similar idea of this, and basically each of the subunit have a hexagonal like distribution pattern. The three kind of like this marks represent the user input of the what we call kind of like the initial seed particle. Basically, you can imagine this is like seed. It will kind of trying to search for its neighbors candidate and trying to evaluate them based on their density. For example, you can see that the others will be accepted as the accepted particles, but this two is kind of like will be excluded. So then those kind of confirmed particles will be kind of like saved in your final particle set, and those accepted candidates will then be the seed particle for your next run expansion. So it's just following whatever in like you specify like the like for your lattice configuration, right? You can then start the second round of your candidate detection, and then you exclude the one that like automatically based on in this case like a cross correlation score. Basically, if you do this iteratively, and you can the auto expansion function like the this algorithm will end when either it's reached to the maximum round number you set, or when it's reached to the edge of your lattice. So then how do we prepare for this expand function, right? So the first thing is, of course, you need some seed particles. So the seed particles can be done in the manual picking module in TomoNet. Basically, you can load all your tomograms in here, and there's those function like picking the top view, bottom view, and side view. So the side view is kind of like easy to understand. It's basically established on the stock init idea. So basically, you use 2 points, like one here, to um, define both the center of your particle, of your seed particle, and the orientation of your seed particle. So what about the top and bottom view? So as you can see, when you go through the higher Z or lower Z area, so you will see a totally different appearance of your particle, right? Because this is like side view, this is top view. So in this case, we could only just use 1 point to define the center, and use the tomogram Z-axis as the particle orientation. Like for the top view, it's in a higher Z, so it's kind of like a oriented in the same direction of the Z, but the bottom view is kind of oriented in the opposite direction of the Z. And then you will save them in the three separate mod file using those imod interface once you click each of them. So, excuse me. So then you can combine all those three views together and do the slug init, and then you basically this is how you generate your seed particles with their coordinates saved in the mod file. The Euler angle will be saved in this init motive list CSV file, and the rotating axis in imode convention is using y-axis as their rotating axis, and this is the link file to the density map for the subvolume extraction. And also there's two kind of like two key elements for running expand function, right? So by doing this, we kind of like get the seed particle, like, locate them. Just we typically want the seed particle to be sparsely distributed over in your lattice when you just like screen your tomograms. So the reason is that it's kind of like a uh, kind of like this is one step, right? This is a two step. So if you all pick your seed particle like that, it's need a lot of a long distance to finally reach to this particle. So which is kind of like a less time efficient. You need to wait more round rounds of like the expand steps to reach to the edge of the lattice. But if you have two kind of like evenly or sparsely distributed over the lattice, you can have you don't need that many round of expansion. So the first part is the seed particle. The second is like we need you use some numbers or parameters to define how to search for the candidates, like the most normally like this vector. We need to define this vector and also a cross correlation threshold to exclude the bad one. So that's kind of like how you use this two kind of like tabs in the ex expand module, right? So the first one is the generate tomograms dot star tab. You basically input your seed particle result folder. So it will generate a star file recording the seed particles from all your like selected tomograms. And this tab is basically mainly you can focus on how to design your lattice pattern. So I highlight this two like key elements. So the transition list is referred to like the vector, a 3D vector from your seed particle center to its neighbors. In this case is 6 of them. So all this measurement should be done within the reference coordinate system. Basically, you also need to input a reference map. They don't need to be really high resolution. They can be kind of like some, you can use the seed particle to do a rough alignment job and get a average from like 50 manually picked particles. Then you can use that as your reference. But basically, after you click the generate, it will gives you a picking.params JSON file format output. With both of this get prepared, then you can run the particle expansion, right? So basically, you just specify your star file, tomogram star file here, and your picking parameter files here, and you can specify which tomograms you want to run the expand and like how many rounds. Basically, it's like how many steps you want to go for your. It really depends on your estimation of your lattice size. If you have a really huge lattice, you can go like 20, 30 rounds. If you have a smaller lattice size, you can use a smaller number. And this is a parameter that's defines the minimum patch size. A patch is a definition of connected or connected is a kind of like a, so for example, this is a small patch, this is a larger patch. So by defining this, giving a reasonable number, you can kind of like exclude the lattice patches that is too small, which is typically we think that's an outlier. And of course you can run this. We have some command recommend option for you. So if you submit your job to the cluster or whatever. Basically, the final outcome will be saved for each of your tomogram will be saved in a final folder. So then you can visualize your tomogram directly here, and basically this is one of the example, like how we use this to pick the VLP like hexamer subunits. So generally, so if we put them together, so basically you need one of the input for the auto expansion function is you need a seed particle information, which is summarized in the tomograms.star file, and you need some search parameter to define the rotational search range, translational search range, your reference, your transition list, which is the center to center vector, and your lattice unit size, cross correlation threshold for excluding bad particles, and you can define how many rounds you expand, and the result will be saved. Like those intermediate results for each round will be saved in the cache folder, and the final outcome, which is the coordinates of your particle, the Euler angle of your particle, and the particle y-axis will be all saved in the final folder. And later you can use some utilities function like here, the P2 star function to export your particles into a Reliant readable format, either in round 4 or round 5. And then you can just keep continuing your 3D refinement or classification jobs in the Reliant. Then the output, it will be a particles.star file. I'll just show you some kind of like example I've been used using this expand function to facilitate my biology study. So this is kind of like you can treat the flagella or the double microtubule as a 1D lattices. By basically just every 96 nanometer they have a repeating unit, this is kind of like some like giant paraflagella rod lattices. It's kind of like a 3D. It's have this crystallized configuration in all three directions. So you can also use that to do the particle picking on the this kind of 3D lattices, and more often is the 2D lattices in the biology world. So basically, one of my major study was the surface layer structure. For example, this one, if you try to use like template matching or whatever, you will end up with a very a lot of false positive on the membrane or on the kind of inner layer or the membrane, since the surface layer here, as you can see, is the outermost layer, is kind of like it doesn't have much feature, and it's just could be easily kind of like considered as a thin layer of membrane or whatever. But if you restrict the kind of like by using the TomoNet expand function, you can kind of restrict the search only on this layer and don't allow it to kind of like jump into far to here or here. So to kind of like introduce those false positive, because theoretically, like they have stronger density than the surface layer. They could be easily kind of like being classified as a positive if just do the global template matching. And from this placeback, you can also see that the particles are correctly picking and they strictly follow the hexagonal lattice pattern, and you can see those domains very well after you average them together. And this is another kind of like an example from our previous work on the N-Hungarian surface layer structure. Basically, we can using this strategy to finally get a 6.4 angstrom resolution of the hexamer subunits, and then by seeing those signal structure, we can using the AlphaFold take advantage of the AlphaFold prediction to kind of get complete one of the pseudo model, atomic model for our structure. Another kind of example is the kind of like VOP capsid particle picking. This is a very kind of like a classic data set, and you can see on the right is the renowned tutorial. Basically, they use this kind of like oversampling on a sphere-like geometry, but for those particles like those one, they're kind of like not strictly follow a sphere-like shape, but using the TomoNet, you can pick them too, and you can tell that using TomoNet, you can actually pick more of those like kind of not that regular shaped lattices. So with that, we can only use like 4 tomograms of this to reach to a 3.2 angstrom subatomic averaging result. And this is kind of like just a kind of like a 3 view of like you basically can picking on any arbitrary shape of as long as it follows a kind of like a lattice configuration. And this is another example of the 2D lattices of this very giant like lattices of nuclear egress complexes. We can eventually also use that to reach to a 5.4 angstrom resolution, and you can see all the helices. So then it's make you easier for some you can go deeper for your structure analysis. And all those placeback things is also being done using the TomoNet placeback function. Basically, you can just input your star file and your average density map and some other related parameters. You can and it can be either Relon 4 or Relon 5 formation. So it will give you a Chimera X command file to basically just load the files in the Chimera X. You could just load the placeback for you. So that's how like that's kind of like the part I want to share for the expansion, and then I just want to talk a little bit more of the neural network picking. So this is kind of like the general design of this neural network based automatic picking. Basically, it's like three section. First, you prepare training data set. For neural network training, you typically want to have a training data pairs, and then you train. We use this UNet architecture as our training model, and then you can do particle coordinates inference by apply this trained model on your tomograms. So I'll break down that in the following slides. So for the training part, it's basically you need to and here you can see there's 2 tabs like training network, predict particles. The training network including 2 parts. One is the training dataset extraction. Basically, you're gonna input a folder that containing the density map and the corresponding coordinates, and then it will be used to train how to extract. So basically, it's encoded the picking as a segmentation task. In a training data set generation, we just kind of like using this just using a 0, 1 mask to represent like the white blocks is kind of like the ones that representing at this location there's a kind of particle positive, and in a dark area it means there's no particle. So then the network will train, okay, so I need to based on this density map, density, we need to kind of like also have a segmentation gives 1 or 0 to represent the appearance of your particle. So it can be done also if you don't want to do this in GUI. There's also a terminal commands for you to do that. It's like extract .py and train picking ts.py, and whatever you input in the interface, the parameter will be saved in a JSON file, which has been kind of like the input of our terminal command, the algorithm. So the second part will be the model inference or the particle coordinates extraction. So you basically input your tomogram and apply your trained model, and it will first give you a predict segmentation map. As you can see, those are the correct positive, like the positive part, right? You can see those little kind of like data bricks, or the white bricks structure represent each of your particles. But of course, there's some outliers like here, are reduced kind of like caused by overfitting or whatever. Then with the output of this, you can apply some. We apply some cluster algorithm to extract the coordinates. Basically, the center coordinates information will be extracted. As you can see in this screenshot, the yellow dot represent the coordinates of your particles, and it can be done using this command if you submit to a cluster. And what I want to highlight, one of the parameters is the what I call a tolerance. It's kind of like you can imagine this is like a regularization parameter which will be either promote or suppress the activate function of those ones or those white things. So which means if you have more tolerance, so it will gives more positive in this which means more white blocks in this segmentation map. But if you have a low tolerance, which means it will lower increase the bar of the activation function to have less of those white blocks or the segmentations, which you probably need to tune a little bit of those hyperparameters for the more satisfied outcome. And for each condition you tried, it will be saved in a separate folder, so you can compare them to kind of like give you a final decision of what conditions you want to use for your final particle picking. And so just wanted to kind of use some kind of like a user case for a quick um, uh, kind of like a demonstration of the implementation for the easiest case is the ribosome, right? So it doesn't need to be a lattices, so it just since it's so isolated and the background is kind of like clean, so you only need kind of like this small uh area of training set with 13 clicks, then you can train this within a minute. Then you can put it in for the whole tomograms. Kind of like the prediction will be done in 1 minute. So with this fast kind of like training and prediction, it will boost up your processing speed when you have hundreds or even thousands of tomograms. And another kind of this is kind of like one of the we've been used in one of our publication on the determination of the LCM glycoprotein structure determination. As you can see, this is kind of one of the um, prediction result. Generally speaking, they're basically done their job, and eventually we can just use this to get a decent result. So I think with that, I think that's probably all I want to share today. This is a acknowledgement. So just thanks, Dr. Zhou, for like supervise this project, and Shiqing and Xingyu are the two undergraduates that work with me closely um, on this software development, and also some support from all the other Zhou labs. And also kind of like want to thank Gonzalo's lab on the Archaea related project, and the Heal lab on the TBRCI flagellum related project, and How to Win lab on the NEC Lattice project. The TomoNet is already been is a open software on GitHub. If you're interested, you can go look into the details in there. So thank you all for listening, and I'll take any questions.
Wonderful. That was a really great talk. Thank you for that. We have two questions in the chat so far, but one of them's a two-parter. So we'll just start with the first one from Alex Stevens. Great talk, Hui. Can you explain how you optimize parameters to deal with curved lattices? For instance, expansion in an icosahedral virus capsid can lead to picks whose Z heights would vary significantly, even just 1 unit out from your picked particle. How do you ensure the extracted box captures this, and how does it impact the parameters you use? For example, do you do fewer rounds of expansion?
Yeah, that's kind of like a very good question, and yes, that could give you a little bit hard time when you do the expansion, but my strategy is the first I will kind of like slide through your tomograms. For example, you mentioned that is the hard part is the top view, right? So that's why in the seed particle generation, in this case, I typically will add a few more top view or bottom view to cover to capture that appearance of your particle, and then limit either increase or decrease the search range for to accommodate the curvature. And also because of the missing wedge artifacts, typically for the top view, you will get lower first correlation score. So to have them correctly picked, you typically also need to first use a lower cross correlation threshold, and then using the other parameters to kind of like clean your particles eventually for the final result. I'm not sure if that answered that question.
If bouncing off of that is, does missing wedge correction in IsoNet not fully recover that distortion that you get along the Z height?
Yeah, it would help. It would help a little bit if you using the isonet corrected tomograms for the picking.
Mm-hmm.
Yeah.
Okay. And then our next question is from Miranda Lynch. For the geometry-based auto expansion on slide 10, how good does the actual lattice structure need to be? Are distortions to the lattice or errors in the lattice structure tolerated by the particle picker?
So I think one example here is for this NEC lattices. Basically, this is just this dimer that form this hexamer, then butt into the ellipsum, like the double layer membrane, and form this kind of like giant vesicles, right? As you can see in this lattice, at this region is kind of a sharp 10, and on the water-air interface is more flat and kind of like spread uh, by the kind of like physical force applied during the freezing. So you can see that even with this kind of like a very a dramatic curvature change, as long as you kind of like define a good range of like either in this like on each axis, if you allow for example, consider that you need to allow for a little bit more uh, angular search, so it doesn't uh, and this will accommodate the defects. For example, here, if it's found some like broken or incomplete part, you just stop picking. So that's how it overcome the defects for the like how good your feature are. I think it can be demonstrated in this example. So I feel like if you look over at this tomogram, you don't see like feature like that. You see a very blurry and very thin layer of that layer. But since you have by using the auto expansion function, you actually have a very strong geometry restraint on this particle. So just trying to avoid like too much search over to some like a very strong density. If that is being kind of like controlled, then you will even though you didn't start with a very good like very strong density, you can still be able to do the picking.
She says, thank you. And she had a part two, but she says that your example answered both her questions. So
Okay.
we really don't need to ask that. And then I have another question about picking the training particles for the neural network based particle picking. How perfectly centered do your training particles need to be? Do they really need to be like directly in the middle of the box in order for it to do a good job picking the rest? Or does it have any like flexibility if you're slightly off center? Because when you're picking by hand, sometimes, if you have a strange shaped particle and you're not looking at the right height.
Okay. Yeah, okay. Yeah, I think that's it can be kind of explained using this slide. So the short answer is you don't have to be perfect centered for your manual picking. In this case, this is just I know ribosome picking is kind of easy, but this is kind of like a show. I just uh try my best just to center but you can see that you can never, when you do manual, you can never
Mm.
perfectly center. You just try your best. Like you don't go off too much, like picking here or picking here, picking here. Otherwise, I think since you do this kind of like in an arbitrary way, so you are not specifically always uh, offset in one specific direction. So I think based on whatever been kind of like trained in the architecture of the UNet, so all those offset will eventually be averaged out and kind of like finally will be converged into the kind of like a real center.
Oh, that's awesome. We have another question. I noticed some missing particles in your lattices. Do you think that these are actually missing, or simply that the particles weren't picked or removed, or they were removed during the reconstruction process? Is there a good threshold to eliminate particles? For instance, if their angle relative to their neighbor is unreasonable? And is there a good way to quantify this so we can justify calling them empty, broken, or something else?
Yeah, I had one slice exactly address, but for the uh, time concern, I removed that slice. But yeah, of course you had that concern sometimes. So the first thing I would try is just go back to your tomogram by using this place back function. So basically, you can see those things, right? Or here, basically you can put your raw tomograms all together with them, and in this case, um, uh, by just directly overlapping the placing back structure on the density, I can see that all the artifacts or the defect are real there, or either I think, let me see if I hide it or delete it. In here, yeah. For example, this one, if you use ISONET correction to, so this is like without averaging, right? Basically, you can see those. So this is the defects region, which is either is kind of like conflicts of the lattice configuration or what, but there's a gap here, and this is the region that is not following the hexagonal lattices, right? You will see also a larger gap here. But basically, yes, you you could use some other kind of like a supplementary function like that to kind of support your observation of those defects. Or uh, yeah, if that's all the question, or did I miss something?
Those are the questions that we have. Does anyone else have any other questions that they would like to ask? You can either drop them in the Q&A section or you can just ask them directly. I think you've explained your project very well, and doesn't seem like there's any additional questions. So if that's the case, then I'd like to thank you on behalf of SBGrid for coming and giving such a wonderful talk today. And it is a very useful tool, clearly, particularly for, I think, structural virologists, but also just anyone doing something with lattices. So keep up the good work. And TomoNet is part of SBGrid for those still on the call. So oh yes, someone's asked, can we get the recording? We will post this recording on our YouTube channel, which is SBGrid TV. So you can go subscribe to us there. And I don't know when we'll post it, but just keep your eye on the channel and you'll see it soon enough. Okay.
Yeah. For anybody who want to try or actually interested, please check our GitHub page, and we have more detailed kind of document there and tutorial. Yeah. Thank you.
Awesome. Thank you very much.